Okay, welcome back to another lecture. Now we're going to talk about some of the molecular mechanisms that actually promote the process of VDJ recombination that we introduced in the last lecture. So there are a lot of molecules, a lot of enzymes that actually help us accomplish this process. So let's get into it. So I introduced in the last lecture the idea that immunoglobulins are encoded by three different loci in the genome, the lambda light chain locus, the kappa light chain locus, as well as the heavy chain locus. And remember that each of these loci encodes a different number of V, D, and J segments depending on the locus that we're in. So, if we want to make a lambda light chain, we need to combine one variable segment with one J segment. How do we actually do that? How does that work at the molecular level? Well, each of these segments is flanked or is juxtaposed in the genome to something that we call a recombination signal sequence. And this is the cue to the enzymes, to the DNA recombination enzymes, that, hey, this is a place where I can actually recombine gene segments together. So what does a recombination signal sequence look like? Well, the recombination signal sequences come in two forms. They're the same thing, but they're two different structures. In general, you can see, so this red box here is one of the segments, like a V segment, that's finishing. But immediately after the J segment, or the V segment, sorry. So immediately after the V segment, we're going to have a recombination signal sequence, or RSS, which contains three parts. There's a heptamer, which is a conserved sequence of amino acids. Conserved meaning we see it over and over again. It tends to not change through evolution. On the other end, there's a conserved nonomer. So heptamer being 7 nucleotide, nonomers being 9 nucleotides. But in between the heptamer and the nonomer, there is a spacer of 23 random amino acids, or nucleotides. We're in the DNA. So 23 nucleotides. And so all they do is just provide some space between the heptamer and the nonomer. Some segments are flanked by an RSS with a 23 base pair spacer, whereas other segments are flanked by an RSS with a 12 base pair spacer. So they look the same for the most part, except there's a different amount of space between the heptamer and the nonomer. And so the reason that we think that it's 12 and 23 is that 12 base pairs is essentially one turn of the DNA double helix, whereas 23 is basically two turns of the DNA double helix. So it seems that this number of base pairs sort of allows the enzymes to sort of dock at the right location and in the right orientation relative to the DNA to actually accomplish recombination. So just a little bit of interest there. So that's why it's 12 and 23. But what's important is that these 12 and the 23 spacers determine which kinds of segments are able to be recombined with each other. So if we look in the lambda locus, for example, the lambda light chain locus, the V lambda segments are always flanked by a 23 base pair spacer recombination signal sequence. So their RSSs contain 23 base pair spacers. Whereas the J lambda segments, they have 12 base pair spacers in their recombination signal sequences. Whereas in the kappa chain, it's flipped. In the kappa chain, the V segments have 12s and the J segments have 23s. And this is important because there's a rule that the enzymes follow here, which is the 12-23 rule. So only segments with 23 base pair spacers can combine with segments with 12 base pair spacers. So that means in the lambda locus, we're only going to get V's combining with J's because they have we're matching a 23 to a 12. Whereas in the kappa chain, it's flipped. And because of this, you can't recombine a V segment from the lambda chain with a J segment from the kappa chain because they both have 23 base pair spacers, and so the spacing just doesn't work. It just, the enzymes can't bring them together effectively. Same thing here. We can't cross combine V and J's from the two loci because of this spacer rule. In the heavy chain, it's pretty similar, except that both V and J segments have 23 base pair spacers, whereas the D segment is actually flanked on both sides by 12 base pair recombination signal sequences. And so here, now V can only bind with, or can only combine with D, J can only combine with D, and they can't combine with each other. And so this effectively makes sure that we are combining the correct segments in the correct order, basically, in the heavy chain. This 12-23 rule is a major way that we make sure that we only end up making functional combinations of the gene segments. So this is an important sort of checkpoint, if you will, in the recombination of these different segments. So, okay, the signals themselves tell us where to recombine, but how do we actually recombine the segments? What does that look like? Well, these RSSs, the recombination signal sequences, are recognized by molecules called recombination activating gene, or they're encoded by genes called recombination activating gene, and so the protein is called RAG. So a combination of RAG1 and 2, they actually bind to the recombination signal sequences in the DNA. So if we look in the germline DNA here, we have the V segment that we're going to use, and downstream we have the J segment that we're going to use. But there's a whole bunch of DNA in between. So how do we get rid of all the DNA in between, which we need to take out in order to actually recombine the two segments that we want to use? Well, the enzyme RAG accomplishes this by binding to the RSS on both the V segment we're going to use and the J segment that we're going to use. And so recombination, or the RAG proteins, are going to bind 123 and 112, as you can see here. And so this is what enforces the rule. So it brings them together, and all of the intervening DNA is kind of looped out on the other end of the RAG complex. The RAG enzymes are going to cleave the recombination signal sequences, and this results in two pieces of DNA, basically. We have what we call the coding joint, which is most of the DNA, which is still intact. But it contains the V and the J segment, or whichever two segments that we're using here. This is the same thing applies to the D and the J combination events too. But for here, we're in the light chain, so we're combining a V and a J. So the coding joint is going to contain the remaining coding sequences, so the actual V and the actual J segments. They're then stabilized by DNA repair enzymes called Q70 and Q80. So that's the coding joint. The other piece of DNA that we have, of course, is all the DNA that we just cut out, and we call that the signal joint. It contains the RSSs, the recombination signal sequences, as well as all of the intervening DNA that we wanted to get rid of. And so it's also then stabilized by Q80 and Q70. An important thing that happens here, though, is we need to bring these two ends back together, right? This won't make a protein. Now we have two broken ends of DNA. So how do we fuse them back together? And for that matter, what do we do with all this extra DNA that's in the signal joint? Well, let's start with the coding joint. So as I said, Q70, Q80, they bind the DNA ends. But what happens when RAG cuts the RSSs out is that it actually forms covalently linked hairpins in the remaining DNA. So remember, this is double-stranded DNA. We're in the genome. So we have a strand of DNA here and its complementary strand here. And their backbones end up being fused together in a covalent structure that we call a hairpin. And the hairpin is closed off. We can't then add new nucleotides to the end of it. So that's a problem. How do we get these two strands of DNA, or these, the two ends of the DNA back together? Well, we bring in more DNA repair enzymes. The next thing that we bring in is a combination of DNA PK and a molecule called Artemis. And they open the hairpin. They break open that covalent bond that's blocking the ends of the DNA, basically. So now that we've opened the hairpins, it's kind of a ragged edge. It's not an edge that we can just like neatly put together. So a few more things happen. The next enzyme in this process, TDT, or terminal deoxynucleotidyl transferase, TDT is fine. It comes in and adds nucleotides. It kind of fills in some holes, and it also just actually adds some random nucleotides to both ends of the DNA. So both to the end of the J segment and the end of the V segment here. But that still isn't great. Right now we kind of just have two open ends with some random nucleotides. We need them to anneal to each other. We need them to ligate, basically, to form a functional strand of DNA. So the next enzymes that come in are DNA ligase, which ligates the DNA based on its name, as well as a partner called XRCC4. What's important here is that it removes any mismatched nucleotides, and it helps the DNA strand find some complementary sequences so that it can anneal, and then it repairs any gaps that are left over from that process. And we're going to go through this step by step in a minute. But ultimately, what results from these three different sort of enzyme complexes is that we form what's called an imprecise coding joint. So we started with our two coding joints, but in the process of all of these different things, we've removed some nucleotides, we've added some nucleotides, and so what we end up with is not simply, if we had taken the V segment and the J segment and smashed them together, right? In that case, that would be a precise joint because the V would end, the J would start, but they would look identical to the way they started. Because we've added some modifications to the junction between the two, now we have what we call an imprecise coding joint. So they're both there, but some slight modifications have been made. Now, though, we have a functional gene, essentially, that contains both segments that we need in order to actually make the variable domain of the immunoglobulin molecule that we're trying to encode for. So that's the coding joint. What about the signal joint? Well, this is a lot more simple, for one, because we don't care as much about it, but the signal joint does not form hairpins when it's cleaved. It actually has 5' phosphorylated blunt ends, and for various reasons, these are easier to just sort of stick back together. So DNA ligase just kind of closes off this loop. Now we have a loop of circular DNA that contains the original RSSs. This is a precise signal joint because we haven't modified it. It contains the recombination signal sequences, basically the way we found them. But now this is extra DNA. For our purposes, it floats away, it gets degraded, and we don't need it anymore. And remember, this is a permanent process. So this DNA is permanently lost from the genome of the cell. And what we're left with is what we're going to use from then on to actually make the immunoglobulin molecule that this B cell is going to create. Okay, so let's look a little bit more about into these processes. I've said a lot of things that happen here. Particular, what are these extra nucleotides that are being added, and why does that matter? So if we go back to our broken coding joint, to recombine in this case, we've changed it to a D and a J segment, but again, this is the same basic process that happens for all the different combinations. We bring together the D and the J segment, and these nucleotides make up their recombination signal sequences. So remember that RAG, that enzyme complex, cleaves off the recombination signal sequences, but it actually leaves a couple of nucleotides behind on both sides. And that cleavage, remember, forms a hairpin, the covalently linked hairpin, which closes off the end of the DNA structure, the backbone, so that nothing new can be added. Well, that's not great. That doesn't actually help us. So the next enzyme that comes in is the combination of Artemis and DNA PK. They open the hairpins, and when they do, the hairpin sort of folds up, if you will. And so these two nucleotides, which were down here before, which were complementary to these two nucleotides, now they come up. And so now we have a sequence of 4 nucleotides instead of a set of matched 2 nucleotides. And so we call these nucleotides P nucleotides because their sequences are palindromic. They're complementary to each other, basically, if you read them out backwards. But these P nucleotides then are sort of form the ragged edges of our coding joints. So how do we get them together? Well, the next thing that we do is we actually add more nucleotides for some reason, although this is important. But for now, this seems a little bit random, but we'll see why it matters in a minute. But the next thing that happens is the enzyme TDT comes in and adds new nucleotides, and we call these N nucleotides. So the Artemis PK complex, Artemis DNA PK complex, it generates P nucleotides, but even more nucleotides get added by TDT, and we call these N nucleotides. So this, it adds a little bit more information here between the D and the J segments. but we still don't have a functionally annealed double-stranded DNA. So how do we get to that? Okay, so we added in nucleotides at the bottom of the last slide. What's going to happen now are those DNA repair enzymes like DNA ligase 4 and its partner, they're going to sort of help fix this now. So the strands are going to be paired, and in particular, the DNA repair enzymes are going to look for any complementary sequences that happen to have been created by the addition of those random nucleotides. And so, it needs to find 2 or 3 nucleotides that match so that they can stick together. In the process, we might have some mismatches. So if we stick these two together, there were nucleotides on either end now that don't match the nucleotide that they're across from. So we need to take those out. We don't want those. But once we find, a little bit of a match to kind of anchor the 2 strands together, then DNA polymerases come in and fill up the empty spaces. And so now we have a nice, complete, annealed and complementary set of DNA in between the original segments. And so this is, why we call it an imprecise coding joint. If we had just put these D and these J's directly in apposition to each other, we might call that a precise coding joint. But we have some extra DNA here, and that extra DNA is coding. It's going to result in extra amino acids being translated between the D and the J segment. And so this process, as we'll see, creates some additional diversity ultimately in the repertoire, because those new amino acids are going to change the tertiary structure of the immunoglobulin molecule, which is going to change which kind of antigens it can bind to. And so this is a major source of extra diversity, as we'll see. But 3 major things happened here. Artemis broke open the hairpin structures, and that created P nucleotides. Then TDT came in and added N nucleotides, and then DNA ligase and its partner came in and kind of fixed the problems that created, repaired the DNA in a way that resulted in a new junctional region between the original 2 segments, which contains both P and N nucleotides. So as I said, the addition particularly of N nucleotides yields additional diversity. In particular, we call this junctional diversity, whereas before, the combination of VD and J segments leads to what we call combinatorial diversity. P and N nucleotides result in additional junctional diversity. So summarizing what we just saw, Artemis cleaves the hairpin, which yields P nucleotides. These P nucleotides are then added onto by TDT with new nucleotides called N nucleotides. Then we bring the 2 strands back together with the help of DNA repair enzymes, and this creates additional coding within the overall gene now, and this contributes to what we call junctional diversity. This is going to make new amino acids, and in the case of immunoglobulin molecules, this kind of only works if we added some kind of triplicate number of nucleotides, because remember that amino acids are encoded by 3 nucleotides each, and so we really need to add either 3 or 6 or 9 or 12 nucleotides total in order to preserve the reading frame of the downstream gene. Sometimes if you have a frame shift, it's fine, but in the case of immunoglobulin molecules, this usually only works if you add a set of 3. So if we think about how this looks, if you stretch your mind back to biochemistry or maybe even general biology, in your DNA sequence, sets of 3 nucleotides form what we call a codon, and this codon is going to determine which amino acid is actually translated by the ribosome. So if we have an insertion mutation where we add 1 or 2 or any kind of non-base 3 number of nucleotides, you shift the reading frame. So whereas before, we had CCC, which made a codon, and ACT, if we just add 1 nucleotide here, basically we're going to change every amino acid after that, because what we call the reading frame has been shifted. So we call this a frame shift mutation. Whereas if we add a triplicate number of nucleotides, so 3, 6, 9, etc., then we're going to add 1 amino acid, but the rest of the amino acids are going to continue in their original sequence. And so normally frame shift mutations really mess proteins up, so we don't want that to happen. And immunoglobulin molecules are pretty complex, and so there's only so many combinations of amino acids that work, and so frame shift mutations tend to create non-functional immunoglobulin molecules. So this process process of VDJ recombination and the addition of N nucleotides often results in immunoglobulins, which are actually not functional. So there's a lot of waste that goes on in this process. However, for those immunoglobulin molecules that do end up working, the net benefit, the net adaptive benefit to us of all of that extra diversity that this process creates is worth all of the metabolic resources that we invest in this process. And so even though we create a lot of junk, B cell receptors that don't work, and those cells end up actually being deleted, we don't keep them around. Even though we put a lot into that, ultimately means that we're be able to mount immune responses to all of the various different things that we are possibly encountering out in the world. So it's a good trade. Okay, so let's summarize all of this. This was pretty molecular, so let's talk about the major themes that we just went over. So gene segments, those V, D, and J segments, are recombined with the help of portions of the DNA that we call recombination signal sequences. So these RSS, these sequences have spacers, which are either made up of 12 base pairs or 23 base pairs. And the rule is that only 12 and only segments with 12 can only bind with segments with 23, or can only recombine with segments with 23, and vice versa. And so this is achieved by enzymes called RAG. And so RAG recognizes the recombination signal sequences and cleaves the DNA at those sequences, resulting in a coding joint which contains the original segments, which code for the immunoglobulin protein, as well as a signal joint, which contains all of the intervening DNA that we need to get rid of, along with the RSSs. The coding joint, which again contains the stuff we want to keep, it requires DNA repair to actually be functional and to actually make an immunoglobulin molecule. So we talked about three kind of major steps. DNA PK and Artemis together, they open the closed hairpin, and that results in the creation of P nucleotides. TDT then adds random extra nucleotides, which we call N nucleotides, and then a combination of DNA ligase 4 and XRCC4. They ligate the two segments back together. They help it anneal, find annealing base pairs that match each other. They take out any mismatches, they fill in the holes, and then we get a nice functional DNA strand as a result. This process of DNA repair adds what we call junctional diversity. So the imprecise coding joint is going to end up containing some random combination of extra P and N nucleotides. And this may add several amino acids, or it may change the entire downstream reading frame. Sometimes that frame shift creates a new molecule that works, but many times, particularly for immunoglobulins, this results in a non-functional mutation. As I've said, ultimately this process is adaptive, even though it's wasteful, because it creates many orders of magnitude more possible, unique immunoglobulin molecules, which underlies that extra diversity we have in our immunoglobulin antigen recognition repertoire. Okay, so that's that for VDJ recombination, at least in immunoglobulin molecules. In the next lecture, we're going to talk about some of the genetics that lie behind how we actually attach the immunoglobulin heavy chains to these recombined VDJ segments that we've been talking about so far. So we'll do that next time.